IB Bio Small Molecules Part 1 will have its focus on providing the fundamental background that will assist your understanding of molecular biology. The essential idea is living organisms control their composition by a complex web of chemical reactions. With this slide, we start Basics 1, atomic structure to bonding. The structure of an atom, as it's best understood for the purposes of studying biology, is a dense nucleus with protons and neutrons, around which move electrons in orbitals of certain energy values. The atom displayed here is helium, with 2 protons and 2 electrons. The inner energy orbital is full with 2 electrons. The electrically neutral atom has an equal number of protons and electrons. Electrons occupy certain energy orbitals. The atom displayed here is carbon with 6 protons and 6 electrons. 2 electrons occupy the innermost energy orbital, while 4 occupy the second orbital. The second orbital can accommodate 8 total electrons. Thus, 4 more electrons would be needed to fill the second energy shell. Atoms can absorb or release energy as electrons are moved from one orbital to another. When atoms absorb energy, electrons are moved to higher orbitals. Think about how light is absorbed during the process of photosynthesis. When atoms release energy, electrons fall back to lower energy levels. Think about the release of energy during aerobic cellular respiration. Atoms that have varying numbers of neutrons are called isotopes. Often, the nucleus of an isotope is unstable, resulting in radioactivity. On the left is carbon-12 isotope with 6 protons and 6 neutrons. On the right is carbon-14 isotope with 6 protons and 8 neutrons. Carbon-14 is radioactive, and its half-life is 5,700 years, the time it takes for half of any sample of carbon-14 to decay. Here's an IB syllabus statement relevant to this introduction to molecular biology. State that the most frequently occurring chemical elements in living things are carbon, hydrogen, oxygen, and nitrogen. The sponge elements are the most abundant elements in living things: sulfur, phosphorus, oxygen, nitrogen, carbon, and hydrogen. Oxygen, nitrogen, carbon, and hydrogen are the most abundant of the sponge elements. Well, despite the fact that the sponge elements are the most abundant, other elements are very important to living things. State that a variety of other elements are needed by living organisms, including sulfur, calcium, phosphorus, iron, and sodium. And can you state one role for each of these elements in plants, animals, and prokaryotic cells? Calcium has an important role to play in the formation of teeth, bones, and shells. It has an important role in muscle activity, nerve activity, and blood clotting. Sulfur has an important role to play in protein structure for all organisms and cell respiration for all organisms. Phosphorus has an important role to play in the formation of DNA and RNA in all organisms, and in lipid membranes. You might remember phospholipids. This would be for all organisms. As well, phosphorus plays a very large role in ATP, which has a role to play in energy transformation in all organisms. Now, work to get to know the roles of these 3 elements and the roles that they play in living things, but certainly as this course progresses, you will strengthen your grasp of this detail, as we will study the biological processes where these elements are important. Iron has a role to play in cell respiration for all organisms, and it has a role to play in oxygen transport in the bloodstream. Sodium has a role to play in nerve activity and water balance in cells. You might remember the sodium-potassium pump. And potassium has a role to play in nerve activity also in water balance in cells. Now work to get to know the roles of these 3 elements and the role that they play in living things. But certainly as this course progresses, you will strengthen your grasp of this detail as we will study the biological processes where these elements are important. Carbon has 6 protons and 6 electrons. The first electron orbital holds only 2 electrons, and we can see that the orbital is full with 2 here. The second orbital holds 8 electrons total. Carbon has only 6 electrons, with 2 in the inner shell. So the second orbital has 4 electrons, and 4 more electrons would fill the orbital. Now, the number of electrons required to fill the orbital determines the number of covalent bonds that the atom will form. Let me say that again. The number of electrons required to fill this orbital, which is 4, determines the number of covalent bonds the carbon atom will form. The number of electrons required to fill the orbital is known as the valence number. The first 3 electron orbitals hold a maximum of 2, and then 8 maximum, and the third shell also 8 maximum. Keep this in mind. For the sponge elements, this table provides the atomic number, the number of protons. The table provides the number of electrons in the outer shell, just the outer shell. And the table provides the valence number, which is the number of electrons required to fill the outer energy shell. The number of electrons required to fill the outer orbital determines the number of covalent bonds that the atom will form. What this means is that hydrogen will make 1 bond, carbon makes 4 bonds, nitrogen 3 bonds, oxygen makes 2 bonds, etc. For hydrogen, 1 more electron is required to fill the first energy orbital. Hydrogen makes 1 bond to fill the first shell at 2. With carbon, the first orbital is full, but the second orbital has 4 electrons. 4 more are required. Carbon makes 4 bonds to fill the outer shell at 8. Nitrogen has 2 electrons in the first orbital and 5 in the second. 3 electrons will fill the second orbital. Thus, nitrogen forms 3 bonds to fill the second orbital. Oxygen has 2 electrons in the first orbital and 6 electrons in the second orbital. 2 electrons will fill the second orbital. Thus, oxygen forms 2 bonds to fill that second energy shell orbital. Here is methane, CH4. The carbon forms 4 bonds, 1, 2, 3, 4, to fill its outer orbital. Each hydrogen needs to form only 1 bond each, 1, 1, 1, and 1, to fill its outer orbital. The electrons are shared to complete the orbitals. 4 covalent bonds. Electrons are shared. The oxygen molecule, composed of 2 oxygen atoms, each of which needs to form 2 bonds to complete the outer orbital, can be seen here. 2 oxygen atoms share 2 electrons each to form 2 bonds. Each atom has a full outer shell of 8. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. Carbon dioxide, composed of 1 carbon and 2 oxygen atoms, can be seen here. The carbon needs 4 electrons to fill its outer shell. Thus, 4 bonds would be formed. 1, 2, 3, 4. Each oxygen needs 2 electrons to fill its outer shell. Thus, 2 bonds each. 1, 2, 1, 2. By sharing electrons in covalent bonds, each atom has a full outer shell. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8 for carbon. Now remember, we're still in Basics 1, atoms to bonding, and here's the next IB syllabus statement of importance. Outline the difference between an atom and an ion. Ions are charged atoms. Atoms that become charged by either losing an electron or gaining an electron. In this diagram, sodium loses an electron to achieve a full outer shell. In turn, the sodium takes on a positive charge, 11 protons in its nucleus, but only 10 electrons total, 2 electrons in the inner shell and 8 in the outer shell. Chlorine gains an electron to achieve a full outer shell. In turn, the chlorine takes on a negative charge. It has 17 protons in its nucleus, but there are 18 protons total. 2, 8, 8. Ionic bonds are formed when ions of opposite charge, positive, negative, come together to form a compound, NaCl. Now we're on to Basics 2, Electronegativity. Electronegativity is a measure of an atom's interest in electrons, a relative measure of an atom's tendency to attract electrons to itself in a bond. Atoms with high electronegativity values tend to form ionic compounds because the grabbing of electrons results in an exchange of electrons, results in ions. Atoms with relatively lower electronegativity values tend to form covalent bonds because the general interest in electrons is less. Thus, the sharing of electrons functions to provide stability. Here's the IB syllabus statement. Define electronegativity. Electronegativity is a relative measure of an atom's tendency to attract electrons to itself in a bond. Electronegativity is an important idea when we study energy transformations, particularly cellular respiration. Stay tuned. Depending on the relative electronegativity of two atoms, say X and Y, electrons could be equally shared as seen in this upper diagram. Bonds formed when the two atoms share the electrons equally are termed non-polar covalent bonds. If the electrons are pulled more strongly to one atom relative to the other, the electrons are unequally shared, as we can see in this second image. The bonds formed in these situations are termed polar covalent bonds because there's slight charge separation in the molecule. If the electrons are outright lost from one atom to another, the bond formed is ionic because we have fully charged species here and here forming the compound, the ionic compound. So, depending on the difference between the electronegativity values of two atoms forming a bond, we can categorize bond formation. If the electronegativity difference is greater than 1.7, ionic bonds are formed. If the electronegativity difference is between 0.3 and 1.7, polar covalent bonds are formed where there is slight charge within the molecule. If the electronegativity difference is 0.3 or less, non-polar covalent bonds form. So I've come back to this table that provides the electronegativity values for certain atoms, and we would use this table to predict the type of bond formed when two atoms come together to form a compound. For example, oxygen has an electronegativity value of 3.5, and hydrogen has an electronegativity value of 2.1. The difference of 1.4 makes a bond between oxygen and hydrogen that is polar covalent. When two oxygen atoms come together to form the molecule O2, the electronegativity difference is 0, 3.5 minus 3.5. That means that the bond between the two atoms of oxygen in the molecule O2 is a purely nonpolar covalent bond. So with water, there are two bonds between the oxygen and the two hydrogens. There's a bond here and a bond here. The molecule is a polar covalent molecule because oxygen has the higher electronegativity value and it pulls these shared electrons towards itself. Thus, the electrons are unequally shared. As a result, the oxygen end of the molecule takes on a slightly negative charge, while the hydrogen ends of the molecule take on slightly positive charge. This is the polarity that's referred to in polar covalent bond. We have a slight separation of charge, negative at this end of the molecule, and positive, slight positive charge at this end of the molecule. Basics 3 involves something called ionization. And ionization is when molecules with polar covalent bonds lose atoms to the solution in which the molecules are found, and thus ions are formed. So for example, some molecules of water lose a hydrogen to become hydroxide with charge, while some water molecules gain a hydrogen to become positively charged. H3O with positive charge is known as hydronium. In this diagram, we can see acetate. The molecule is here, and it has a polar bond right here between the oxygen and the hydrogen dissociates. The acetic acid then takes on a negative charge as the hydrogen is released, and the hydrogen is positive charge. By releasing the hydrogen ion, increasing the hydrogen ion concentration in the solution, molecules like acetate would be called acidic. Basics 4, a quick look at acids and bases. An acid is a substance that increases the hydrogen ion concentration of a solution. So hydrochloric acid is an acid simply because this hydrogen ion dissociates in a solution. This increases the hydrogen ion concentration in the solution, thus we would call HCl an acid. A base is a substance that reduces the hydrogen ion concentration of a solution. When we have ammonia in solution with excess hydrogen ions, the ammonia picks up the hydrogen ion to become NH4. This is ammonium. And in reducing the amount of hydrogen ion in solution by picking it up, we have a less acidic solution, or in other words, we have what's called a base. Another example, sodium hydroxide is a base simply because it dissociates in solution. This hydroxide then picks up the hydrogen ion from the solution, reducing the hydrogen ions in solution. The hydroxide and the hydrogen ion together form water. In this diagram, we can see ammonia picking up the hydrogen ion from solution, effectively reducing the hydrogen ions in the solution, so ammonia would be considered a base. And basics 5, the last of the basics, is one last thing to think about as we move forward with molecular biology, and that is molecular shape. Shape is very important to function, and you should always be thinking about the shapes of molecules, particularly their three-dimensional shape. On the left is a flat two-dimensional representation of methane, CH4. I've showed you this image earlier in the movie, but on the right is the actual shape of methane. It's actually a tetrahedron. In addition to shape, it's important to realize that molecules can move and rotate. Single bonds allow rotation. Notice how there's been a slight shift seen in this molecule ethane. Ethane is a hydrocarbon, and again, it's important to remember that molecules can have slight shifts in shape. Double bonds, if they exist, are shorter and they allow for less rotation. As you think about three-dimensional shape, I want you to consider that there are tight spaces and open spaces and grooves. In other words, there are nooks and crannies of molecules that are important to the interaction of molecules with other molecules. For example, molecules being acted on by enzymes. In this diagram, we see the amino acid alanine, and it's easy to forget the complex shape of a molecule as seen here on the left when looking at two-dimensional representations as seen on the right. Keep in mind the three-dimensional quality of all molecules, and that shape and nooks and crannies are very important for molecular interaction. I want you to realize that slight differences in shape and structure can make for huge functional differences. Estrogen and testosterone are both synthesized starting from cholesterol, so they both have a similar four-ring structure. But the subtle differences in chemistry result in dramatic differences in function. Estrogen increases female qualities, while testosterone increases male qualities. And that brings us to the end of Small Molecules Part 1, an introduction to some of the fundamental aspects of molecular biology. In Small Molecules Part 2, we will focus on water, structure, and function.